So first of all, thanks a lot for the invitation in the most beautiful city in the world, I believe. I had been the chance to do the military service in this city at Le Cascine because I was an officer of Aeronautica Militare, so Italian Air Force. And so every time I come back here, I am very happy. So let's try to discuss about rational design for radiation-targeted therapy combination, because there are no more targeted agents, and when they invited me, all the topics were covered. So I will try to do a methodological presentation. Those are my disclosure. So what is really essential is to develop preclinical studies in order to guide the development of new treatment combinations. So I was listening before to the concept that maybe CDK4/6 inhibitors may induce a cell cycle arrest, and so will induce a reduction of the activity of radiation oncology. I am not so sure about this. I really believe radiation oncology together with targeted agents has a huge opportunity. We need to generate more data, both on the preclinical setting and also in the clinical setting. And since now we have in breast cancer and in other disease many targeted agents, now it's mandatory to do something like this. So it's time to work together and to generate data. We were discussing with Hope a few minutes ago, and as she said, if you look on the PubMed, on a prospective trial combining TDM1 plus radiation therapy, there is absolutely nothing. That is incredible. So why not working together to generate data? So when we design clinical trial, the first endpoint should be safety. This is not a bias because I work in a phase 1 unit. On the long term, I do not expect any detrimental effect of combining targeted agents with radiation oncology. But what we need to understand is really in the context of a combination, if we need to perform a due dose escalation study. The dose escalation can be on the radiation oncologist part, but can be also on the targeted agent part. Modulation of the dose is quite common in breast medical oncology. Just think about the NATALY trial, in which a dose reduction was performed in the adjuvant setting, rather than 600, that is the dose registered for the metastatic setting. In the adjuvant, there is a 400 milligram dose of CDK4/6 inhibitor. In my opinion, many drugs currently available are overdosed. So we should start from the principle that many doses registered are overdosed. You know perfectly that with ribociclib, you have exactly the same activity with 400 milligram in terms of median PFS and response rate. So if we start from this assumption, it will be very easy to design clinical trial. So we need to discuss a new clinical trial design, specifically time to event continual reassessment method, to design phase 1 and phase 2 trial before moving, of course, in a potential combination. And what is essential is to provide a clear pathway in this study design. So you discussed before about how many trials are ongoing, targeted agents and radiation oncology. And I found this paper published on Radiation Research, just to understand the characteristics of proposed clinical trial combining radiation therapy plus targeted agents. So I classified them by cancer type. So very common, of course, 17% on brain mets. Brain mets seems to be your obsession. In any case, in brain mets, in non-small cell lung cancer, in breast cancer, you have so effective treatment today that I really believe we can delay radiation therapy. So I don't need to do a trial of trastuzumab deruxtecan plus radiation therapy, neither of lapatinib or tucatinib with radiation therapy, because systemic treatment is very effective. So we need to replicate the model of lung cancer. You know, in lung cancer, actually, you delay radiation therapy, because when you have an oncogene addiction, tyrosine kinase inhibitors or ADCs can be more effective than radiation therapy. Then you have, of course, trials, very few, 5%. It's very few number. 5% of trial for breast cancer, then we have cervical, GI, lung, prostate, sarcoma. Other trials are related to the extent of the disease, and 41 trials are related to concurrent radiation therapy and targeted therapy. Then we have metastatic disease, 23%. Recurrence of disease is a very common clinical presentation. We have many local recurrence in breast cancer. So it's a huge opportunity to design clinical trials on this setting. And then we have also not specified. Regarding phase of the study, look, 45% of them are phase 1. It means safety analysis. Then you have phase 1-2, 16%, and then, of course, as expected, some phase 3. So discussing about approach to preclinical studies in the letter of intent proposal, this is an analysis of trials submitted to NCI, you have many of them that are related to preclinical design. So preclinical model and histology match. Look, they are 75%. And when we discuss about molecularly targeted agents, here we have the surprise. So we have 4% angiogenesis. Then we have 6% on target of apoptosis. DNA repair are 30%. So I am imaging PARP inhibitors plus radiation therapy. That is, of course, a critical question, at least also in breast cancer. 45% of them, as expected, are on immunomodulation. Then we have mitosis, protein expression, and metabolism. But where are targeted agents? This is the topic of our meeting. So it seems that there are very few trials combining radiation oncology plus tyrosine kinase inhibitors or antibody-drug conjugated or potentially also antibody. So the proposed radiation therapy trial, of course, is here, and then also some preclinical studies. In blue, you have fractionated, and in orange, you have single dose. And this can also extrapolate the concept of adjuvant and metastatic setting. So it's essential to understand that there is a huge potential to combine radiation therapy, not only for radiosensitizing cells, because the concept is not to radiosensitize, but to have a synergistic effect. Of course, in immunotherapy, this is very easy because I expect that radiation therapy can work as a boost for immunoactivation. So I am really excited when I see some large trials in the past using vaccination therapy plus radiation oncology. Don't forget that in allotransplantation, a whole body radiation therapy is mandatory. So it's very clear the correlation between radiation oncology and activation of the immune system. Looking at the cell model studies, look at here, you have many trials with PARP inhibitors. Then many of them are also together with anti-angiogenic agents, and very few, finally, with other agents that should be used, finally, to deposit high level of energy and to increase activity of radiation therapy. So there are many challenges to combine radiation therapy with targeted agents, specifically in the adjuvant setting. First of all, regulatory guidance. If you look at all the trials in the adjuvant setting, with the exception of few, just the ones with pembrolizumab in the adjuvant setting or trastuzumab in the adjuvant setting, usually the advice of FDA is not permitting concurrent administration of abemaciclib or ribociclib with radiation therapy. Also in the Olympia trial, using olaparib was not permitted to give olaparib plus radiation therapy. So, of course, from a regulatory point of view, this is an impasse that you can potentially challenge, because you can ask the regulators, okay, we complete the clinical trial, and then we do a tail with 50, 60 patients, exploring a dose escalation of the drug concurrently with radiation oncology. And so this is essentially to be done, and it's very easy. I believe any company will be available to spend a little bit more of money to generate data in this context. So, first of all, let's sit down together and let's try to change the draft guidance and to ask in any trial a tail of the patients exploring the combination of radiation therapy plus the targeted agent with a dose escalation. This is possible. It's not impossible. Everything can be done if you discuss with regulators. You have complex models because in the majority of the cases, the drug is developed in a preclinical setting just without exposure to radiation therapy. We need to understand if the synergistic effect can increase some types of toxicity, but we need to redefine the endpoints. Endpoints cannot be only safety with the drug alone, but can be also safety in combination with radiation therapy. So it's a research collaboration, finally. Which are the challenges? Because, of course, anytime we have to design a trial, we have challenges. First, limited relevance of preclinical work. In most of the cases, there is absolutely nothing. Then the differences between early phase trial design with and without radiation that usually are substantial. So in all the phase 1A design, it's not permitted to give a concurrent radiation oncology. Also, if you deliver it between one cycle and another one. And response rate is not a useful surrogate of activity in radiation combination trials, because you can give also a response with radiation therapy alone. So first, we need to demonstrate the potential efficacy. So does the drug radiosensitize tumor cells with a specific concentration? Then we need to explore the mechanism of radiation modification. So don't you believe that after radiation therapy, something can change in the tumor microenvironment and so potentially can reduce the absorption of the drug or can increase the absorption of the drug? We can define a target or a signature that defines which tumors are best radiosensitized or have a best synergistic effect. And this today is possible because we have radiomics. And I am very surprised about the potential effect of radiomics in the next years. We need to investigate scheduling issues. Maybe a continuous administration is not permitted, but an intermittent one, yes. So there is a lot to discover. We need to investigate normal tissue response. What is going on a normal tissue if you concurrent give a targeted therapy plus radiation therapy? And we need also to discover potential new biomarkers. So the clinical success of many targeted agents finally is based on an oncogene addiction. There is no phase 1 with a targeted therapy without a biomarker, with the exception of CDK4/6 inhibitors. But in many phase 1, you have a target, you have a drug. And these also should move from the concept of precision medicine to precision radiation medicine. I like that Philip is very convinced about this. So precision radiation medicine should also rely on genomic information of the tumor. And many groups tried to develop algorithms like this. So just to remember to you, in the United States, there is a cancer imaging archive that is led by the NCI. And they established a prospective randomized trial that is called PRISM, that includes service for managing radiology and pathology image with artificial intelligence. So the key objective is to open access information repositories for early and advanced cancer and to generate knowledge in terms of prediction of response. The knowledge, of course, is essential because this will permit us to integrate clinical data and radiomic data and to generate algorithms that may predict activity of radiation oncology and not only of targeted agents. And this is a quantitative imaging and omics analysis. So you integrate radiomic, genomic, proteomic in patients who received a specific radiation treatment or a specific targeted treatment. It's a very complex project. It has been published 3 years ago on JCO Clinical Informatics. But as you can see over here, there is really an application programming interfaces can integrate big data and can generate a lot of data. So the collection, the number of the patients, then radiology imaging, pathologies, and recently also genomics. I believe this is an effort that is really essential for the future because now technology can give us the opportunity really to change the clinical practice. I am a strong believer that artificial intelligence will give dramatic changes in the way we do trials. This is an ontology-driven somatic research. So if you integrate all the data, pathology, genomics, radiomics, you can generate algorithm predictive of response. And we need also to understand that according to the targeted agent, you have a really different type of response. The response generated by a chemotherapy is completely different from the one generated by a tyrosine kinase inhibitor or an immunocheckpoint inhibitor or a CDK4/6 inhibitor. So RECIST is not enough. You need really to have different type of assessment of response according to the treatment we are delivering. And I was very impressed when the Gustavo Roussy published this trial. This is the first radiomic signature that may predict activity of immunocheckpoint inhibitors. This was the radiomic workflow, so imaging selection, delineation of volume of interest, so exactly like you do when you do radiation oncology, imaging processing, and then extraction with the clusterization of outliers, so exceptional responders and progressor. And this finally generated a radiomic signature. Radiomic signature that can be predictive of response to immunotherapy or progression to immunotherapy. And this was exactly the data. Look, the performance of these radiomic signatures was then validated in prospective trials. And the difference is outstanding. So some radiomic features can predict an excellent outcome or a poor outcome after exposure to immunocheckpoint inhibitors. This can be done by you in 24 hours, I believe. And you can generate radiomic signatures of response or better outcome for breast, lung, and any other type of disease. So what's about immunomics and radiomics? This is really impressive because just using imaging, you can predict the outcome of patients. And the radiomic signature was also associated to a different biological environment of the tumor, many CD8 infiltrating tumors. So imaging is more than picture. They are data. So it's really essential to understand that once we perform an intervention with radiation therapy, targeted agents, you have an imaging before and an imaging after that can generate predictive models. This is the simple concept I would like to give to you. We need to use imaging to better understand also the interaction between radiation oncology and targeted agents. So how to design clinical trials? This is the last part of my talk. The first question is the choice of systemic agents. This is a key question, of course. So are we going to design a trial with ADC, with CDK4/6 inhibitors, with tyrosine kinase inhibitors? So we need to know the pharmacokinetic, and for the majority of the agents we know perfectly. We need to know the penetration into tumor type, and you have this because in the preclinical studies you have pharmacodynamic studies for any single agent, and you can know the effect into the tumor and in the microenvironment. Is systemic side effect profile understood? Does toxicity overlap with radiation oncology? Does agent have single agent activity? Do we have any subtypes where the targeted drug knows better? What's about radiosensitivization? And is there evidence for normal tissue radiosensitivization? So these are the key questions we have to ask when we design trial together. The other point is the choice of the tumor type. And here is very simple. It's breast cancer. So you know everything about radiation oncology in breast cancer and targeted agent in that setting. And then we have to design the optimal methodology to deliver this data. Radiation doses, volumes, fractionation schemes, quality assurance control, because not in all the centers are so good in delivering radiation oncology, incorporation of novel functional imaging endpoints. I believe this is essential. And incorporation, eventually, of pre- and mid-treatment biopsies. This is not usual, but maybe, at least in the metastatic setting, you should do. You need to consider pragmatic solution for speeding up drug development in combination with radiation therapy. And we need also to establish an extant standard of care for radiation therapy trials, because many times in multi-institutional trials, you have a different way of delivering radiation therapy. The trials, in my opinion, should be organ-specific, or at least disease-specific. I don't believe we can do an agnostic trial when you combine in a disease a targeted agent. And the primary endpoints should be acute toxicity. And toxicity should remain the main endpoint, because the question you are asking me is, can we deliver TDM1 together with radiation therapy? So it's very easy to design a trial like this. With 50, 60 patients, you complete the trial, and believe to me, you go on JCO or on annals, because this will be an important trial for clinical practice. And in some circumstances, you define a maximal dose based on toxicity, because if we consider a dose escalation, we can modulate the targeted agents, starting from the minimum effective doses, increasing the dose. And we need, of course, to plan the trial to utilize the preclinical data. This is the only way, but this is possible because we do this in phase 1 trials. So in conclusion, for many common cancers, adding novel targeted agents to radiation therapy or radiochemotherapy, in my opinion, may increase cure rates. We need only to study this. And scientists, I believe, should not overly rely on in vivo model. We need really to design clinical trial in the clinical setting. Thank you.